Hello friends, welcome back to my YouTube channel Nasrin Padasali. So today we are going to study about the cell inclusion bodies. So inclusion bodies are commonly present in all kind of bacterial cell. So these inclusion body are formed by aggregation of substances, and these substances are either they might be organic in nature or inorganic in nature. These inclusion bodies are clearly visible under light microscope and phase contrast microscope. The location of these inclusion bodies is that they are present in the matrix of cytoplasm. So next is regarding to classification of inclusion bodies. Inclusion bodies are classified into organic inclusion body if the substrate present is organic in nature, and inorganic inclusion body. Example of organic inclusion body include Cyanophysynchronic granule, Carboxism and gas vacuoles, whereas example of inorganic inclusion bodies include polyphosphate granules and sulfur granules. Next, the location of inclusion bodies or regarding to structure. Inclusion bodies, some inclusion bodies lie free in the cytoplasm, whereas some inclusion bodies are enclosed by a shell, and the thickness of shell will be 2 to 4 nanometer in thickness, and the shell will be a single layer membrane structure which might be composed of proteins and phospholipid. Example of inclusion body which lie free in the cytoplasm include polyphosphate granule, Cyanophysynchronic granule and Glycogen granule, whereas example of inclusion body that are enclosed by a shell include polyhydroxybutyrate granules, Glycogen, sulfur granule and carboxism and gas vacuoles. Okay. So next, what might be the function of the inclusion body? So generally, inclusion bodies are used for storing carbon compound, inorganic substances and storing energy. In addition to that, few inclusion bodies may reduce the osmotic pressure, and some other inclusion bodies play a vital role in movement of bacteria. So first inclusion bodies is a storage inclusion. So the most common type of storage inclusion include glycogen inclusion, PHA, that is polyhydroxyalkanoate granules, sulfur globules and polyphosphate granules. Okay, so some inclusion bodies are observed only in certain group of organisms. Like one example is Cyanophysynchronic granules, which is present only in cyanobacteria. So the first we will studying about this PHA granule. So generally carbons are stored in this PHA granule, and several type of PHA granules are being identified, but most commonly studied one is a poly 3-hydroxybutyrate, that is PHB. Okay, so this PHB contain a hydroxybutyrate molecule which are joined to the ester bond. Okay, so ester bond is present between carboxyl group of one molecule and hydroxyl group of another molecule. So the bond present is ester bond. Next, this PHA granule are surrounded by a single layer shell of protein that are made up of a high amount of proteins and a small amount of phospholipid. So one example of this PHA granules is a biodegradable plastics or biopolymer. So the next inclusion bodies is with regarding to the sulfur granule. Okay, so this sulfur granule is used by some bacteria in order to store sulfur temporarily. One example of bacteria is photosynthetic bacteria, which generally use this hydrogen sulfide as the photosynthetic electron donor. So normally bacteria will be employing water as electron donor, but photosynthetic bacteria will be using this hydrogen sulfide as a electron donor. Okay, and once they had reduced the hydrogen sulfide, means they will be accumulating the resulting sulfur either in periplasmic space, or they might be storing them in a special cytoplasmic globules like sulfur granules. Okay. So the next inclusion bodies is glycogen. So glycogen is a kind of organic inclusion bodies, and glycogen, the structure of glycogen is that they are a polymer of glucose subunit which are composed of long chain made up of alpha 1,4 glycosidic bond, and the branching chain are connected to them by alpha 1,6 glycosidic bonds. Okay, both of this glycogen and PHB are the inclusion bodies that generally they are used for carbon storage reservoirs, thereby both are involved in providing material for energy as well as biosynthesis for few cellular constituents. Next inclusion body is polyphosphate granules. So many bacteria store their phosphate as a polyphosphate granule, which is also called as oleutin granules. Okay, so this polyphosphate is nothing but they are the linear polymer of orthophosphate that are joined together by ester bonds. Okay, and thus oleutin granules, which is also called as this polyphosphate granules, they function as a storage reservoir of phosphate. So phosphate is an important cellular constituent that are present in nucleic acid. And this particular polyphosphate granules is sometimes called as metachromatic granules because these granules have a capacity to show metachromatic effect. So what does mean by this metachromatic effect? When bacteria that consist of this polyphosphate granules are stained with dye like methylene blue or toluidine blue, the bacteria which containing this particular polyphosphate granule will be appear either as a red or as a different shade of blue. So the next thing is regarding to Cyanophysynchronic granule. This Cyanophysynchronic granule is present in cyanobacteria, and this Cyanophysynchronic granule they composed of a large polypeptide chain and they consisting of approximately equal amount of amino acid. That is, this particular Cyanophysynchronic granules consist of large polypeptide which are made up of the large amount of amino acid arginine and aspartic acid are highly present in this particular Cyanophysynchronic granule. And this particular Cyanophysynchronic granule we can easily observe them under a light microscope. So what is the function of this particular Cyanophysynchronic granule? They store extra nitrogen that are available for bacteria. Okay, so this Cyanophysynchronic granule are involved in storing nitrogen. So next particular type of inclusion bodies is microcompartment. These microcompartments are unique in nature, and these microcompartments serve a function other than simply storage substances. Okay, so previously we just mentioned storage inclusion, but this microcompartment, they have unique function. Okay, this microcompartment, they generally have they are relatively large polyhedron which are formed by one or more different, so they have a structure similar to virus, and these microcompartment currently they are called as metabolic compartment. Okay, and inside this microcompartment they will be carrying out a specific series of reactions will be carried. Okay, and then generally these microcompartment or general function is that they either enhance a metabolic pathway or either protect a metabolic pathway from an attack of few any other inhibitors. Okay, so they enhance or protect the key metabolic pathway that are occurring inside a bacterial cell. So these are a few examples of microcompartment. So ethanolamine utilizing microcompartment, which is called as EUT. So generally gut bacteria will be often have a potential to form this particular ethanolamine utilizing microcompartment because ethanolamine is abundantly present in the intestine. Okay, so this ethanolamine is a breakdown compound of this phosphofatidyl ethanolamine. Next type of microcompartment is propane diol utilization microcompartment. In short form, they are called as PDU. Okay, so this PDU is a proteinase as well as their subcellular structure that serves as an organelle for the metabolism of this 1,2 propane diol. So this particular microcompartment is present in an organism called Salmonella enterica. Okay, and this particular microcompartment encapsulates several related enzymes. Okay, so what are the enzymes that are required for metabolism of this 1,2 propane diol? Then definitely all enzymes will be present inside the particular microcompartment itself. The last type of microcompartment which we are going to study in detail is this carboxisome. So carboxisome, generally they are present in either cyanobacteria, and in addition to that they are present in few other carbon fixing bacteria. So these carboxysomes are polyhedral in structure and their size is 100 nanometer in diameter. And inside this carboxysome microcompartment there is a presence of enzyme called ribulose 1,5 bisphosphate carboxylase. In short form it is called as ribisco. And this ribisco play an important role in the CO2 fixation. So what is mean by the CO2 fixation? So CO2 fixation is a process of converting the carbon dioxide absorbed from the atmosphere into sugar. So this carboxysome is acting as a site of carbon dioxide fixation. So the last type is that some inclusion bodies are playing an important role in the movement of bacteria. Okay, so two type of examples or two type of inclusion bodies are playing an important role in the movement of bacteria include gas vacuoles and magnetosomes. So first we'll be studying about gas vacuole. So gas vacuole, they are present in many photosynthetic bacteria, and in addition to that they are present in aquatic prokaryotic. So example of aquatic prokaryotic include your halobacterium, which is a salt loving archaeon, and another bacteria is a Thiotrix, which is a filamentous bacteria. So in this particular bacteria gas vacuole is present. So gas vacuole generally contain a small and hollow and cylindrical structures called gas vesicle. So this gas vesicle, the specialty is that they are impermeable to water, but they are freely permeable to atmospheric gases. Okay, so the function of this gas vacuole is that these gas vacuoles generally regulate the buoyancy to float. So in this aquatic prokaryotic, the presence of this gas vacuole, they provide a buoyancy to float at a depth that is necessary for the proper light intensity, oxygen concentration as well as nutrient level. So that is the function of gas vesicle which is present in aquatic prokaryote, and they help the aquatic microorganism to float at a particular depth in a marine environment, which is very much favorable for providing the nutrient level and oxygen concentration. So the next bacteria is with regarding to this magnetosomes. So magnetosomes, magnetosomes is nothing but they are the inorganic inclusion bodies that can be used for purposes other than storage. Okay, so aquatic magnetotypic bacteria will generally use this particular structure in order to orient them in an earth magnetic field. So those bacteria which using this earth magnetic field, they will be present, they will be having this magnetosome with you. Okay, so these magnetosomes are nothing but they have the intracellular chain of this magnetite particle, that is Fe3O4 particle, and each of this ion particle is considered as a tiny magnet. So while studying this particular state, keep your mind concentrated on magnet. Okay, so you know what is the function of magnet. Okay, so the next is with regarding to this particular this magnetosome, that is northern hemisphere bacteria will be using their magnetosome chain in order to determine northward and downward direction, whereas southern hemisphere bacteria, that is those bacteria that are residing in the southern hemisphere, will be using this magnetosome in order to orient southward and downward. At the same time, this magnetosome is used by the bacteria in order to find a place where there is an enough amount of nutrition rich food is available. So magnetosome is also used by the bacteria for food survival also. Okay, next, for the cell to move properly within the earth magnetic field, this magnetosome they need to arrange in a form of chain. Only then bacteria will have a capacity to orient themselves in a magnetic field. Okay, so for this particular function, a special cytoskeleton protein called MAMK is being identified, which has been providing a framework upon which a chain of magnetosome will be forming. So this is how the magnetosome will be, friends. So here you can able to see here the cytoskeleton framework will be there. So there MAMK will be, MAMK which is special protein will be there, and on that protein magnetosomes will be formed. Okay, so this is present inside the bacteria. So I hope this particular video will be really helpful, friends. If it is really helpful and if you want few other topic notes means, then please do tell me in the comment section. I'll be definitely making you this PowerPoint presentation and I'll be uploading you. Thank you, friends. Thanks for watching this video. If you didn't subscribe to my YouTube channel, please subscribe to my YouTube channel.